Okay, here, so now it works. Okay, the gentleman here is Leonardo da Vinci, self-portraits from young to old, and the question we're asking, how can you prevent this young gentleman becoming so old? We view aging as a deterioration of biological networks, where you start as an ordered network, and then it becomes disordered, damaged, and then essentially you die. And what my lab focuses on is we try to find small molecules that extend lifespan and slow down this process. And then by figuring out what those molecules do, we hope also to learn something about aging. So I'm going to apologize immediately to the evolutionary biologist showing such a simplistic view, but essentially aging occurs in a selective shadow and is a consequence of neglect to select. So what you see here are survival curves from humans, chimpanzees, worms, and flies. They depict the fraction of individuals alive at a certain age, at different time scales, years for humans, and chimpanzees, days for worms and flies. Now let's superimpose mortality, which essentially tells you the probability to die at a certain age, and not surprisingly, that mortality goes up dramatically. And if we now superimpose fertility data, we see that when mortality is low, when fertility is high, mortality is low. And the consequence is that with the decreasing fertility, we get a decreased selective pressure to maintain the organism. So in a first approximation, I would say nature did not optimize survival after reproduction. Now, from a drug discovery point of view, that's actually good news, because we only have to improve something that nature didn't bother to really improve. If nature spent 4 billion years to improve something, then good luck making it better. So how bad is aging and how much space is there for improvement? What you see here are the mortality data of humans from 1999. Blue are men, red women, black is the total population. These show deaths per year per 100,000 at a given age. So if you're a human and you're born, you're actually quite prone to death. That probability starts to drop until you're about 15 years old. At the onset of reproduction, for reasons that I don't think are understood, mortality goes up to quite a bit. And then the most interesting point to me is here, where mortality stays actually stable for about 10 to 15 years. So you can keep mortality stable. So if we just hypothetically could say, okay, if we could stabilize mortality at that rate for the entire life of a human being, we would end up at about 690 years, or an 8-fold increase. So aging essentially costs each of us, with this exponential rise in mortality, about 600 years. Now that's not a completely harebrained number. Is seen here in C. elegans. These are survival curves of C. elegans. C. elegans is a small nematode that lives about for 3 weeks, shown here with N2, but there are two actually interventions. H1 is one of them that make the animal live 8 times as long. So I'm not suggesting that we're going to achieve this in humans, but the point I want to make is that if we can improve this part of the curve just by 1 to 2%, that would actually result in about a 10-year increase in lifespan. Here again, this part optimized by nature from the ordered network becomes disordered, damage, and death. And in these selection shadows, where basically all of the age-associated diseases start to dramatically increase. Now I'm going to tell you, as I said, we do drug discovery, we do large-scale screens for compounds that extend lifespan, and then we use them to figure out what they do and learn about aging. So I'm going to tell you about 2 small molecules that we found, and they seem surprising to act along the dogma of molecular biology, for information paradigm from DNA, RNA, protein, and then some other steps that lead to organismal life. And as we heard, aging progressively deteriorate any of those molecular components. And so I'm going to talk about Mianserin, which is an atypical antidepressant that protects at least the C. elegans from aging, and Minocycline, which protects the proteome from aging. So this is all going to be C. elegans based research. So Mianserin is an atypical antidepressant. It targets serotonergic and adrenergic receptor, and it extends lifespan in C. elegans, which is shown here. So here we have a survival plot. So you have the, this is the control animals, and then if you treat them with, this is the fraction of animals alive as a function of age. You can see they die, live about 3 weeks. If you add Mianserin, you get a 40 to 50% increase. Now, genetically, we have previously shown that that requires serotonergic signaling, but that never explained really what's actually happening and how does inhibiting serotonergic signaling increase lifespan. And to look at that, we did a very simple experiment. We essentially asked what gene expression changes with age. So we took adult worms day 1. So we do all experiments essentially in adult worms. And then day 1 adults, and then day 3, day 5, day 10, we extracted RNA from these different cohorts and did an RNA-seq. And we saw about 3,000 to 6,000 genes that changed expression. And we started to do gene set enrichments, which is shown here. So each of these triplicate pie charts here represents a gene ontology. It's not so important what's written above here. And this is the gene expression changes on day 3, 5, and 10. In white are genes that don't change. In blue, genes that go up, and in black, genes that go down. What we noted that we have over 250 of these, and what is common with them is that the fraction of genes that change into opposing directions seems to increase with age. Now, if these are functional units, modules, you would expect them to be you would expect that the stoichiometry between them to matter, which would suggest that them going into different direction with age is actually a deterioration of the transcriptional network. And it is not a change intended with a specific function. To look at that, we came up, we called this phenomenon transcriptional drift, and we essentially came up with this plot where we normalized gene expression of every gene relative to the expression in the young organism, where essentially with the logic that mortality is lowest then, so that's the optimal situation of a network, and chronic and permanent changes away from this is probably problematic. And then instead of looking at individual genes changing, we look at groups. So these are, for example, lysosomal genes, and use essentially error bars to quantify the change in stoichiometric relations from these genes with each other. So you can see that this is worm data on day 1, 3, 5, and 10, and you can see that the transcriptional levels of these genes seem to go off in different directions the older the animal gets. And you can do this kind of plot with every group of gene that you really want. So here, for example, it's done for the entire transcriptome. It's the elegans. This is the variation in stoichiometry that you see between animals of the same age. And then when you compare them along lifespan, you see there's a dramatic increase in transcription drift. The same is true for mice. These are, I forgot mouse, I think they're brain data. And then here is human data from the frontal cortex. You can see for the first 60 years, not much is going on in the human brain, at least in terms of aging. And then at the age of about 60, right when cognitive problems start to set in, transcriptional drift starts to take off. But the most important argument we have to say that that's not a functional change, but that's actually deterioration, is that when we treat the animals with Mianserin, the drift is attenuated, which is shown here. So here's again transcriptional drift for control animals. And when we treat them with Mianserin, we see that by day 10 animals have the same drift as untreated 3-day-old animals. And what that suggests is essentially that Mianserin either prevented the transition from an ordered to a disordered state, or reimposed order when it was added. And to test that, we did a simple experiment. We took worms again and we added Mianserin on day 1, 3, 5, or left them untreated, and then extracted RNA on day 10 and looked at transcriptional drift. And that's shown here. So this is on day 1. This is when we added just water on day 1 as a control, and this is transcription drift on day 10. If we added Mianserin on day 1, we see a massive suppression of this. If we add it on day 3, we see less of a suppression. And if we added Mianserin on day 5, which is when it's already in a somewhat disordered state, we see actually a slight increase in transcriptional drift. And that correlated perfectly with the lifespan data. So this is the control lifespan. If we added Mianserin on day 1, we have lots of suppression of drift and we have a big increase in lifespan. If we add it on day 3, we still have a 15% increase in lifespan, but less suppression. And if we add it on day 5, we even get a slight reduction in lifespan consistent with a slight increase in transcription drift. And what that said essentially is that what Mianserin does, it prevents this transition from an ordered to a disordered network. And in fact, we looked at over 3,000 animals, and this shift here is actually a perfect parallel shift in the Gompertz lifespan curve. And yesterday we heard about these different shifts, and I would bet that a lot of these shifts will be accompanied by a reduction of transcriptional drift. So so these animals are actually longer young, and you can really see that if you look at the phenotype. But from a drug discovery point of view, that was actually rather disappointing, because it says we show that Mianserin doesn't work when you add it to old organisms. If you want to use small molecules or that geroprotectors to treat age-associated disease, they all happen in the disorder damage step. So we want to find molecules that are still capable of increasing lifespan when the animals are already old. And so we first thought nature's way to treat damages through stress responses. We asked, can we find a stress response that can be activated in old organisms that we could use, then activate with a small molecule, and then to make the animal live longer. So we did a very simple experiment again. We took worms, we added that reported on different stress responses. We added stresses on day 1, 5, and 8, and looked at the stress response. And the results are shown here. Top is oxidative stress response, ER unfolded protein response, and heat shock response. Each of these is 100 worms in a 96 well. And these are worms that become fluorescent when the oxidative stress response is activated. And here we quantified the different responses. So you can see for all the stress responses, oxidative stress response, ER unfolded protein response, and heat shock response, there's a dramatic decline in the ability of the organism to mount a stress response once they became old. And which essentially told us, and we looked at that in terms of drift, is that this disorder here actually inhibits stress responses. The activation of stress responses in aged animals, which is probably why diseases start to creep up when we get older. But the problem was that by day 8, essentially, we couldn't find any stress response that was still activatable when the animal were already old. So to solve that, we decided, okay, let's screen for molecules that still can increase lifespan on day 8, and then find out how the molecule is doing that. So what you see here is the life history of a worm. Starts as an egg, goes through 4 larval stages, and then becomes a reproductive adult. And normally we always added the drug when we screened on day 1, but this time we decided to add it on day 8. And the results are shown here. So this is the change in lifespan. These are 21 drugs. You can see they all increase lifespan when they are added on day 1, but most of them fail to increase lifespan when added on day 8. For example, here is Mianserin that I showed you before, which even had caused a significant reduction, a slight but significant reduction in lifespan. The only compound that we found that still worked was Minocycline. It's a tetracyclic antibiotic. Actually, for the worm aficionados here, worms eat bacteria. We had to do this experiment with killed gamma irradiated bacteria to make sure that the drug really works on the worm and not on the bacteria. So you can see if you add minocycline on day 1, you get about a 40 to 50% increase in lifespan. If you add it on day 8, that's much reduced, but there's still a sizable increase of 22%. In mammals, minocycline is also an anti-inflammatory. There have been a number of potential mechanisms suggested, none of which we spent 2 years chasing them, but none of them was able to explain how that works. So what we did is we did chemoproteomics to actually identify where minocycline was binding, and that is shown here, the cytoscape plot. Essentially, it's a translation, peptide metabolic process, ribosomal assembly, which is essentially the ribosome. And the ribosome makes proteins. So we asked if we it did a pulse labeling experiment. So we treated worms with water or minocycline, and then looked at incorporation of N15 labeled amino acids. So in black is the water-treated animals, in red are minocycline treated, and each dot is a protein in black if it comes from the water treated, in red from the minocycline treated one. You can see that there's about a 25% reduction in protein synthesis when we treat the worms with minocycline. We did an entire series. We also did radioactive labeling, polysome profiling, and right now we're doing ribosome. And so, but what that gave us the idea is, so yes, yesterday we heard a lot about the proteostasis machinery, which folds and degrades proteins. And so the biggest load on this proteostasis machinery is newly synthesized proteins. And if they're not properly folded, they start to form aggregate, which is the basis of many neurodegenerative diseases like Parkinson's, Alzheimer's disease, fatal familial and insomnia, and so forth. And so the idea was that if we reduce minocycline, the translation by about 25%, we should generate folding capacity and we should improve protein folding. And we tested that in worms that express a fluorescently labeled alpha-synuclein, which is shown here. So these are heads of worms that express a fluorescent alpha-synuclein, which is a protein that aggregates in Parkinson's disease. And you can see that with increasing age here, day 8, 11, 16, 19, the staining becomes more and more punctate, suggesting protein aggregation of alpha-synuclein. And when treated with minocycline, that doesn't happen. We did a series, we also did this in HeLa cells, and we did a series of other experiments. And one of my favorite one, however, is shown here. So this is HSF1 mutant. These are again survival data from C. elegans. The HSF1 mutant has a mutation in HSF1, which is the main transcription factor for the chaperone system in C. elegans. And these animals have serious protein folding problems and are short-lived because of that. And they are actually very hard to make live long. Most longevity mechanisms can't extend lifespan of HSF1. However, if you add minocycline, you can almost double the lifespan extension, or you get more than double the lifespan extension of these animals. And in the end, they actually live longer than wild-type untreated animals. So why does minocycline work in aged organisms while most others don't? It is shown here. So you have translation that produces some unfolded proteins. Occasionally there occur misfolded proteins which activates a stress response, which then by E1F2 alpha phosphorylation inhibits translation. In aged organisms, as I've shown you, these stress responses no longer come on and so and fail to activate the so-called integrated stress response, which and prevent this feedback loop that under normal circumstances always in young animals prevents the translation machinery to produce too many unfolded proteins. So this feedback loop is broken in aged animals. So minocycline very simply fixes that, but just attenuating translation slightly. We've shown, I'm not representing the data here, but we've shown that minocycline does not require the integrated stress response and can inhibit translation even in the absence of E1F2 alpha phosphorylation. So here is the summary. So we think about aging from an order, disorder, damage, and death perspective. We have Mianserin that seems to block the transition from an ordered to disordered state. We have minocycline that indirectly generates folding capacity by reducing protein synthesis. And this nicely goes along the molecular information paradigm that's necessary for organismal life. And obviously we're looking now for molecules that block part of these downstream mechanisms, see whether they can actually add it even later in life. And here, acknowledgement of the people that did all this work. And I hope I stayed in time and I'm ready for questions.